IB Cell Biology Part 2 will continue with some of the overarching themes of cell biology, including the use of microscopes, the size, scale of various cellular components, and the surface area to volume relationship. The essential idea, once again, is the evolution of multicellular organisms, allowed cell specialization, and cell replacement. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused here. Biological study includes molecules, cells, tissues, organs, organ systems, individuals, populations, and the interaction within their environment. Here's the first IB syllabus statement of this movie. Compare the magnification and resolution of electron microscopes and light microscopes. Light microscopes seen here have a relatively low magnification, not exceeding 1,000 times magnification. They also have a low resolution. Resolution is a measure of the ability to distinguish 2 points as separate points. For light microscopes, 2 points would need to be relatively far apart in order to be distinguished as separate. But light microscopes are inexpensive as they use glass lenses, and they use visible wavelengths, which allow specimens to be seen in motion and in color. Here's an image of some of the earliest known microscopes. Remember that maximum magnification would be 1000x, and the distance between 2 points would need to be relatively far in order to be distinguished as separate points. This means low resolution. Here's a photograph of an electron microscope. Electron microscopes have high magnification, extending to 250,000x, and high resolution. 2 points can be very close together and still be seen as separate. Light microscopes use mirrors to focus visible wavelengths. Electron microscopes use magnets to focus electrons with very short wavelengths. The use of very short wavelengths in electron microscopes allows for high resolution and high magnification. This is an image of a nucleus within a cell. The image is magnified more than 10,000 times with an electron microscope. Here's a comparison of the electron and light microscopes. While the electron microscope allows for high magnification, and the distance between 2 points in order to be resolved is very short, this is high resolution, the light microscope allows one to view living specimens in color. In other words, the light microscope allows one to see motion in natural colors. Here's the next IB syllabus statement. Compare the relative sizes of molecules, cell membrane thickness, viruses, bacteria, organelles, and cells using appropriate SI units. Molecules are measured in nanometers, 1/1000th of a meter. Membranes are 10 nanometers thick. A virus is 100 nanometers. And prokaryotes, bacteria, are 1,000 nanometers or 1 micrometer. 1 micrometer is a millionth of a meter. Cells with a nucleus, eukaryotes, are 10 micrometers up to 1,000 micrometers. Notice the range of structures that can be seen with the electron microscope and the range of structures visible to light microscopes. You can see that the sequence of each structure relative to the one previous to it increases by a factor of 10. Factor of 10, factor of 10, and so forth. Study this slide. Here are some metric conversions that are important for you to know. 1 millimeter is 1,000 micrometers, and 1 millimeter is also 1 million nanometers. 1 micrometer is 1,000 nanometers. This micrograph is looking at large molecules known as ribosomes. If we use this scale, we could estimate a single ribosome, to be approximately 25 nanometers. Remember, molecules are measured on the scale of nanometers. In this micrograph, we are looking at 2 cell membranes, side by side. We can use this scale line to estimate the width of the membrane at 10 nanometers. Membranes are layers of molecules, as we will soon understand, and it makes sense that membrane width would be measured on the scale of nanometers. A virus is a non-living biological particle seen in this micrograph and also seen in this drawing. A virus is approximately 100 nanometers in length. In this photograph, we can see a bacterium, and using this scale line, we can estimate the length of the prokaryotic cell to be approximately 1 micrometer, or 1,000 nanometers. Organelles, such as a mitochondrion seen here, is a structure that lies within larger cells, and it has the same size as a bacterium. Mitochondria and chloroplast as internal structures are thought to have evolved from formerly free-living bacterial cells. Thus, the similarity of mitochondrial size with bacteria. This is not a coincidence. The chloroplast seen here is an organelle, and like the mitochondria has the same approximate size to a bacterium at 1 micrometer, or maybe just a little bit larger. Chloroplasts are larger organelles than mitochondria. The eukaryotic red blood cell is one of the smallest eukaryotic cells at 10 micrometers. Eukaryotic cells have a nucleus. The paramecium is a single-celled eukaryotic organism, approximately 500 micrometers in length. Eukaryotic cells range from 10 to 1,000 micrometers, although there are cells longer than 1,000 micrometers, like muscle cells or fungal hyphae, as you might remember from part 1 of this series. The human egg cell seen here is 1,000 micrometers, which is the same as 1 millimeter. Notice the numerous sperm cells, and they're significantly smaller at 50 micrometers long. Again, here's an outline of the range of sizes of various cellular structures. I'll leave this slide for you to study. Here's the next IB syllabus statement. Explain the importance of the surface area to volume ratio as a factor limiting cell size. As we approach the relationship between surface area and volume, keep in mind that the surface, or the cell membrane of cells, regulates the entry and exit of materials, while the volume, the cytoplasm of cells, determines the rate of metabolism. The rate of heat production of a cell is a function of volume, while the rate of exchange of materials with the environment is a function of surface area. As a cell gets larger, the surface area to volume ratio decreases, and we can see that here. The cell model on the left has 2 by 2 by 2 dimensions, resulting in a 3 centimeter square to 1 cubic centimeter ratio, while the cell model on the right with larger dimensions of 4 by 4 by 4 has a surface area of 1.5 centimeters square to a volume of 1 centimeter cubed. As the cell grows in size, the surface area does not increase as much as the volume. Thus, exchange of materials becomes limiting relative to the metabolic needs of the cell as determined by its volume. Shape also influences the surface area to volume ratio. Cells that are more cubed, less elongated, have a lower surface area to volume ratio compared with cells that are long and slender in shape. Notice that both of these cell models have exactly the same volume, but the long, slender model has a much higher surface area per volume ratio. In this diagram, you can compare the surface area to volume ratio of the small cell to the surface area to volume ratio of the large cell. I'll let you do the mathematics given the dimensions of a side here with these two cell models. But the obvious question then is, how are large organisms adapted to the surface area to volume problems when size increases? And the answer is that organisms become multicellular. Cells stay small, but organisms can become large by being multicellular. And that brings us to the next IB syllabus statement. Explain that cells in multicellular organisms differentiate to carry out specialized functions by expressing some of their genes but not others. Remember that multicellular organisms have differentiated, specialized cells, the epithelial cell, the nerve cell, the muscle cell. All the cells have the same complement of DNA, But differentiated cells have certain regions of the DNA that are expressed, while other regions of the DNA are not expressed. Cells that are differentiated have different functions within the larger body of the multicellular organism. The IB asks that you be able to calculate the magnification of drawings and the actual size of structures and ultrastructures shown in drawings or micrographs. I want to emphasize drawings and micrographs. This statement does not refer to estimating the size of structures while using a microscope. This statement refers to calculating the magnification of drawings and micrographs that have been published, that are on a piece of paper. Keep this in mind. So let me reiterate. The calculation of the magnification of structures, ultrastructures, and micrographs involves published images. These images are available in books or online sources, and they will provide some information along with the image. Keep this in mind. So my question is, using this scale line, how would you calculate the magnification of this micrograph? So here's the challenge. Calculate the magnification of this published micrograph of a mitochondrion. You're going to use this scale line and a ruler, get out a ruler, so that you can calculate the magnification of this image. Here's the formula. Magnification is equal to image size over actual size. I call the formula the MIA formula, and the equation can be rearranged to find actual size if you're given magnification. All units need to be the same for actual size and image size, so you will need to remember some metric conversions. 1 millimeter is equal to 1,000 micrometers, or 1 centimeter is equal to 10,000 micrometers. So back to the mitochondrion, the easiest approach is to use a ruler to measure the length of the given scale line. Let's measure this as 1.5 centimeters in length. That's equal to 15,000 micrometers. I'll let you work on the math. This is your image size. The actual size is given, 1/10 of a micrometer. Divide to calculate the magnification as 150,000x. Remember, both values must have the same units, and they cancel, so that this is magnification. It's unitless. Now, you might have been tempted to calculate the magnification of the entire mitochondrion, and that's fine, but realize that everything in the image is magnified equally, so using the scale line is the easiest approach. But had you decided to use the entire mitochondrion, the measured length with a ruler would have been 15 centimeters, thus your numerator, the image size, 150,000 micrometers. The estimated length of the mitochondrion using the scale line would be 1 micrometer, because the scale line fits approximately 10 times across the length of the mitochondrion. Realize that the same magnification is calculated whether you use the scale line or the whole structure for the calculation. Remember, units must correspond in order to calculate the correct magnification. So here's a slightly different question. Calculate the actual size of structures and ultrastructures shown in micrographs. The actual size is asked for, not magnification. In this case, magnification is given. Once again, measure the structure, say the length of the bacterial cell, by putting a ruler up to the screen. This is about 16 centimeters in length. So we're going to rearrange the equation to solve for actual size rather than magnification, as the magnification was given as 100,000x, and the numerator is 16 centimeters put into micrometers, and that's going to be 160,000 micrometers. We divide 100,000 into 160,000, and we get 1.6 micrometers. Remember, bacterial cells are approximately 1 micrometer in size. Here's another chance to calculate the actual size of this nucleus in this micrograph. Given the magnification of 40,000x, get your ruler out and measure the width of this nucleus as approximately 8 centimeters. Again, we've taken the MIA formula and rearranged it to calculate for actual size. We know that magnification was given as 40,000x, and we know that the numerator at 8 centimeters would have 80,000 micrometers in place. Thus, the actual size of this nucleus is 2 micrometers. Or I could have asked you to calculate the magnification of this published image, this cell nucleus, and with a ruler you would have measured this as 8 centimeters. I would have given that the width of this distance in the micrograph was 2 micrometers, and you would have had 80,000 micrometers over 2 micrometers, and that's 40,000 x. Again, the actual size across the image was 8 centimeters or 80,000 micrometers. The actual size of that line was given as 2 micrometers, and we divide to get 40,000 x. Notice that because the units here are the same, they cancel. You're left with a unitless value for magnification of 40,000 times magnified. And that brings us to the end of Cell Biology Part 2.